Number 1. The Madygen Forest. Picture yourself standing at the edge of a Central Asian Rift Valley lake roughly 230 million years ago. The canopy above is a tangle of broad-leaved ginkgos, cicadophytes, and slender Voltsia conifers. The horsetails of the genus Schizoneura grow in dense thickets at the water's edge. Towering above them, the Lycopsid Pleuromea rossica spears the humid air. Something flickers between two trunks overhead. It is not a bird, it is not a bat. It is Chaerovipteryx mirabilis, a tiny diapsid reptile gliding on skin membranes stretched between its enormously elongated hind limbs and its tail. The membrane catches the warm air. The animal drops silently across the canopy gap. And then, somewhere in the still black water below, another one falls in. This is the Madygen Lagerstätte. The deposit sits in the Fergana Valley of southwestern Kyrgyzstan, in the Madygen Formation, dated to the Ladinian to Carnian stages of the Middle to Late Triassic, between 237 and 227 million years ago. The Soviet paleontologist Alexander Sharov led the principal excavations through the 1960s and 1970s, and the site has since produced more than 20,000 cataloged specimens. The killing mechanism was the lake. These were stratified Rift Valley lakes in an actively subsiding basin between two closing oceans. The bottom waters were anoxic, sometimes hypersaline, and the chemocline was razor sharp. Any animal that fell through it sank into a chemically inert grave where decay bacteria could not function. The shale that resulted is finely laminated black rock that preserves insect wing venation at microscopic resolution. The companion glider Longisquama insignis, described by Sharov in 1970, carried paired rows of elongated dorsal, scale-like appendages. A minority view in the Russian school argues these are true proto-feathers, placing avian origins outside Dinosauria entirely. Mainstream cladistic analyses reject the claim. A 2006 description added Kyrgyzosaurus bukonchenkoi, a lacertilian-like reptile with skin impressions intact. Marina Sherbakov of the Russian Academy of Sciences has cataloged more than 200 insect species across 14 orders. The site preserves the oldest known mole cricket and the oldest crane fly. The freshwater shrimp Triops madygenicus is nearly identical to living tadpole shrimp, showing 230 million years of morphological stasis. The basin itself collapsed during the Carnian pluvial episode around 234 million years ago, ending Madygen as a death trap. What remains is one of the strangest forests ever fossilized, and almost nobody outside paleontology knows it exists. Number 2. The Geiseltal Lignite Forest. If you could walk into a Middle Eocene swamp 45 million years ago in what is now Saxony-Anhalt, you would not last long. The air would be heavy with methane and hydrogen sulfide. The ground would be a floating peat mat, soft enough that a misplaced step might drop you waist-deep into anaerobic mud. Tannins from the leaf litter would stain the water the color of tea. The pH at the bottom of the bog would be somewhere between 3 and 4, acid enough to dissolve a calcium phosphate skeleton over time. That is exactly what happened. The Geiseltal Formation sits in the Geisel River Valley near Brownsbitru and Mechelen, exposed during open-cast lignite mining between 1933 and 1993. The deposit dates to the Lutetian stage, between 48 and 41 million years ago, with the richest layers near 45 million years. More than 50,000 specimens were cataloged before the mines closed. Over 40 mammal species, 12 reptiles, 11 amphibians, 50 bird species, and thousands of insects came out of the coal. The bog dissolved bones but preserved everything else. Feather barbs, hair, skin, gut contents, and chlorophyll-containing leaf cuticles survive intact. Maria Voigt's 2019 paper in Papers in Paleontology documented the famous Eopelobates frog orgy layer, where male and female frogs were trapped in mating amplexus and preserved together. The horse ancestors Propyliotherium and Lophiotherium, both fox-sized, preserve stomach contents of grape seeds and laurel leaves, providing direct dietary evidence. The crocodilians Diplocynodon and Alognathosuchus document year-round warmth across the Eocene. The site yielded the holotype of Eurohippus mesalensis, a pregnant horse ancestor with the fetus still inside her uterus, the umbilical cord and amniotic membrane preserved. Maria McNamara and Patrick Orr's 2013 melanosome analysis revealed that some Eocene beetles retained their iridescent green and bronze structural colors 45 million years later. The trap mechanism was simple. Animals stepping onto the floating peat mat sank through into deeper anoxic mud, where the bog's chemistry inhibited bacterial decay almost completely. A minority interpretation by Jens Lorentzen in 2018 argued some mass mortality events were tied to volcanic gas releases from nearby vents. The hypothesis remains controversial. The mine sites are now flooded as the Geiseltal Lake's recreation district, sealing the unexcavated material underwater. What remains in the hollow collections forms the most complete Eocene mammal sequence in Europe, a permanent record of a forest that quietly digested everything that fell into it. Number 3. The Eckfeld and Enspel Maar forests. A Maar lake looks innocuous from above, a small, perfectly circular pond ringed by subtropical forest, the water surface mirror flat. The trouble is, what is dissolved in the deeper water? Periodically, when the chemical stratification destabilizes, the lake can release a cloud of carbon dioxide that suffocates everything passing low overhead. This is precisely what happened at Lake Nyos in Cameroon in 1986, killing 1,700 people and 3,000 head of livestock in a single night. 44 million years ago, in the Eifel volcanic field of what is now Rhineland Palatinate, the same process killed bats and primates and small horses on a smaller, slower, more relentless scale. The Eckfeld Maar formed around 44.3 million years ago by phreatomagmatic eruption, the precise date confirmed by Mertz and colleagues in 1991. The Enspel Maar in the Westerwald formed in the late Oligocene, around 24.8 million years ago, dated by Sanidine argon-argon analysis by Mertz and Rhynie in 2005. Mars form when rising magma encounters groundwater, producing a violent steam explosion that excavates a circular crater that subsequently fills with stratified water. The Maar profile is the death trap. A narrow circular outline, steep walls, and a deep anoxic monomictic bottom water layer, rich in hydrogen sulfide. Any animal that drowned in the surface water sank through the chemocline into a mud where decay bacteria could not work. Eckfeld preserves the holotype of Eckfeldia rauhi, a primitive primate. The site also yielded marsupials, early rodents, lemurs, bats, and even a chameleon, all in laminated bituminous shale. Herbert Lutz of the Mainz Natural History Museum described mummified pregnant Propalaeotherium horses with intact fetuses in his 2007 paper. The Eckfeld bat Paleochiropteryx was preserved with stomach contents of moths and lacewings identified to genus level. Enspel yielded Eomidae rodents with hair impressions including Eomes, alongside the swimming pangolin relative Eurotamandua. Christoph Stefaniak and colleagues at the Senckenberg Research Institute documented gas bubble lesions in Eckfeld vertebrate skeletons consistent with rapid death by lake bottom carbon dioxide release. Hans Schall in the 1990s had argued the Mars were ordinary glacial kettle lakes, a minority view since rejected by the chemical and stratigraphic evidence. Occam-Reisdorf's 2012 study examined post-mortem buoyancy, concluding that internal gas rotates corpses ventral side up before sinking. The Eifel volcanic field includes more than 200 craters, and the system is dormant but not extinct. Lacher C last erupted around 12,900 years ago. Future eruptions remain entirely possible. Number 4. The Joggins fossil cliffs. Of all the death traps in this list, Joggins gives you the most literal image. Animals fossilized standing upright inside the hollow tree stumps exactly where they died. The Joggins cliffs run for 15 kilometers along the southern shore of the Cumberland Basin on the Bay of Fundy in Nova Scotia. The site became a UNESCO World Heritage Site in 2008. The fossiliferous strata span the Pennsylvanian subperiod, dated between approximately 318 and 313 million years old. Charles Lyell visited Joggins in 1842 and 1852, describing the upright tree stumps in his Principles of Geology. His second visit was accompanied by William Logan and John William Dawson. It was Dawson, on that second expedition, who discovered the first vertebrate remains inside hollow lepidodendron tree stumps in 1852 including the holotype of Hylonomus lieli, the earliest known reptile. Hylonomus was just 20 centimeters long, with a skull only 2 centimeters across. It is the oldest unambiguous member of the Amniote that gave rise to reptiles, birds, and mammals. The Lepidodendron and Sigillaria stems were hollow because the trunks were largely filled with parenchymatous pith that decayed quickly after the trees fell or died standing. The stump stood upright in the forest floor when the surrounding peat swamp flooded. Sediment then filled the hollow chambers around any animal that had fallen or crawled inside. Robert Carroll documented additional Joggins specimens in his 1964 paper, including the larger amphibious tetrapod Dendropedon acadianum, which also became trapped in upright stumps. The trap stump hypothesis was challenged in a 1996 paper by Mike Calder and Howard Falcon Lang, who argued instead that animals were swept in by flash floods. A 2010 study by Falcon Lang's team rebutted the flood hypothesis by demonstrating in-life articulation of skeletons within the stump fills, supporting the original entrapment scenario. The trap functioned because rising water in the swamp could enter through the stump base, while trapped vertebrates could not climb the smooth interior walls. Joggins preserves more than 96 successive forest horizons, each representing a temporary ecological succession on accreting deltaic sediment. The forest grew in equatorial Pangaea around 5 degrees south of the Paleo-equator, with continuous high humidity and rainfall. The site also preserves Arthroplury, the giant millipede-like myriapod, which reached 2.6 meters long, leaving distinctive paired furrow trackways called diplicnites. Carboniferous atmospheric oxygen peaked around 31%, the highest in Earth's history, allowing the gigantism seen in Joggins arthropods. The Bay of Fundy's enormous tidal range of up to 16 meters continuously erodes the cliffs, exposing new fossils but destroying any specimen not quickly extracted. Number 5. The Clarkia Fossil Forest. The single most unsettling thing about Clarkia is what happens when you pull a fossil leaf out of the sediment. For about 90 seconds, the leaf is bright green. You can see the venation. You can see the chlorophyll. Then, as the leaf encounters atmospheric oxygen for the first time in 15 million years, the green fades. Within 2 minutes, the leaf has oxidized to brown and then to black. The color was preserved in anaerobic mud for 15 million years and destroyed by 90 seconds of fresh air. The Clarkia lakebed sits near the town of Clarkia in Shoshone County, northern Idaho, exposed in a road cut on State Highway 3. Charles J. Smiley of the University of Idaho investigated the site systematically, beginning in the early 1970s. The deposit is dated to the late Middle Miocene, between approximately 15.8 and 15.5 million years ago. The lake formed when basaltic Columbia River flood basalts dammed the ancestral St. Maries River drainage, creating a stratified lake roughly 20 kilometers long. The sediment is fine varved clay with annual laminations between 1 and 3 millimeters thick, allowing season-by-season climate reconstruction. More than 130 plant species have been identified, including magnolia, quercus, acer, liquidambar, ginkgo, sequoia, and metasequoia. The diversity surpasses modern temperate North American forests. Insect fossils preserve compound eye facets, wing membranes with original color patterns, and gut contents recoverable in dilute hydrofluoric acid. Edward Golenberg's 1990. Nature paper claimed DNA recovery from magnolia leaves. The result has been contested as likely contamination by modern DNA, and most subsequent workers considered deep-time DNA preservation at Clarkia unproven. A 2018 study by Stephen Manchester and Hongshan Wang documented the leaf fossils at high resolution, identifying new species of acer and tilia. The preservation mechanism is anoxic bottom water below the lake's thermocline, combined with high tannin content from rainforest leaf litter. The lake's stratification was permanent due to its depth of at least 70 meters. Fish swimming into the lake's anoxic deep zone suffocated within minutes and sank to chemically inert mud, where decay halted. Ralph Taggart used Clarkia pollen sequences in 1989 to document the Miocene climatic optimum cooling and the subsequent shift to drier conifer-dominated forests. Brent Lindwall and Walter Lewis interpret the chlorophyll preservation as magnesium substitution stabilization, within the porphyrin ring rather than simple absence of decay, a chemically unusual pathway. The site is publicly accessible at the P.F. Smiley Fossil Bowl, where visitors can dig for and keep leaf fossils for a small fee. Number 6. The Rhynie Chert. If you want to picture the Rhynie Chert in its prime, imagine a Devonian wetland in what is now Aberdeenshire, Scotland, around 411 million years ago. The plants are small, leafless, stoloniferous things less than half a meter tall. The animals are arthropods. There are no land vertebrates yet. Hot springs rise through the wetland, depositing silica that hardens into glassy rock. Then something snaps. A new pulse of silica-rich water surges out of the spring vents at near boiling temperature. It impregnates every plant cell it touches, every arthropod leg, every fungal hypha. Within hours, an entire living ecosystem is fossilized at cellular resolution. The Rhynie Chert outcrop was discovered in 1912 by William Mackey while mapping the Old Red Sandstone. The deposit is dated to the early Devonian Progean to Empsian stages, with a precise radiometric date of 411.5 million years from Mark and colleagues in 2011. The Chert preserves the oldest known continental hydrothermal paleo spring deposit and one of the oldest preserved terrestrial ecosystems on Earth. The plants include Rhynia gwynvaugani, Aglaophyton major, Asteroxylon mackii, Horneophyton linieri, and Nothia aphylla. Andrew Knoll and Keith Stewart have documented nuclei, chloroplasts, and even mitotic figures inside individual silicified cells. The animal fauna includes the trigonotarbid arachnid Paleocarinus, the springtail-like collembolan Rhyniella precursor, and the predatory chelicerate Palaekia octoniza. Lyle Anderson's 1995 census identified more than 20 species of arthropods in addition to the plants and fungi. Rhynie also yielded Rhynieognatha hirsti, originally interpreted in 2004 by Engel and Grimaldi as the oldest known insect with mandibles suggesting flight-capable Pterygoda. In 2017, Joe Haug and colleagues reinterpreted the specimen as a non-insect myriapod. The debate is still active. The fungal mycorrhizal associations at Rhynie are the oldest direct fossil evidence of mutualistic plant-fungus symbiosis, documented in detail by Tim Taylor of the University of Kansas. The site preserves complete plant life cycles, gametophytes, sporophytes, and intermediate developmental stages of the same species, an unmatched resource for early land plant biology. The hot spring chemistry derived from rhyolitic volcanism associated with the closing of the Iapetus Ocean and the Caledonian orogeny. Modern analogs include the Yellowstone geyser basins and Iceland's hot springs, where silica precipitation can fossilize plants within weeks. The Rhynie animals breathe atmospheric oxygen through tracheae, with book-lung structures preserved in some trigonotarbids, providing the oldest direct evidence of how early land animals processed the air around them. Charcoal layers in the chert indicate that wildfires burned across the early Devonian landscape, the oldest preserved fire records on land. Number 7. The Gilboa Forest. Most of the forests in this list killed the animals walking inside them. The Gilboa Forest killed an entire ocean. The site sits near the town of Gilboa in Schoharie County, upstate New York, discovered during dam construction along Schoharie Creek for the New York City water supply in 1920. The deposit dates to the Middle Devonian Gevetian stage, approximately 385 million years old. It is the oldest known forest preserved with in situ standing tree bases. Winifred Goldring and colleagues at the New York State Museum excavated more than 200 fossil stumps between 1920 and 1925. Many were later destroyed by reservoir flooding. The stumps were originally interpreted as cycad-like seed plants. William Stein and colleagues reinterpreted them in 2007 in Nature, describing complete specimens of the basal cladoxylopsid tree Watiza. The trees grew to at least 8 meters tall, with slender trunks and branched crowns of leafless photosynthetic axes, resembling a giant tree fern more than a modern tree. A 2012 re-excavation by Christopher Berry and William Stein revealed lycopsids and N-neurophytalian progymnosperms growing alongside Watiza, creating a multi-layered canopy structure. The forest at Gilboa was the first to develop deep root systems penetrating more than a meter into soil, fundamentally transforming continental weathering. The root systems are preserved as the genus Aeroerytherium, with axes up to 15 centimeters in diameter branching dichotomously underground. The consequences were planetary. Deep rooting caused biological weathering of bedrock, and a massive drawdown of atmospheric carbon dioxide. Global concentrations dropped from around 2,000 parts per million to roughly 300 parts per million across the Middle and Late Devonian. The nutrient runoff from the new forests fertilized ocean surface waters, triggering algal blooms whose decay produced widespread bottom water anoxia. The Kellwasser and Hengenberg extinction events at the Frasinian-Femmenian boundary around 372 million years ago eliminated approximately 75% of marine species. Thomas Algeo of the University of Cincinnati has been the primary advocate of the forest extinction hypothesis since his 1995 paper with Stephen Scheckler in Philosophical Transactions of the Royal Society. Hans-Peter Schultke's 2003 minority view argued that some Gilboa stumps belong to an unrelated taxon distinct from Watiza, a claim partially supported by 2017 anatomical work. Modern parallels include the recognition that coastal mangrove deforestation accelerates coastal eutrophication, mirroring the Devonian feedback loop on a much smaller scale. The forest killing of marine animals was indirect but global. The atmospheric and oceanic consequences of land plant expansion were severe enough to be classed as a contributing driver of the late Devonian mass extinction. Number 8. The Florissant Redwood Forest. 34 million years ago, a redwood forest in what is now central Colorado stood at the foot of an active volcanic center. The redwoods themselves were the close relatives of the giant sequoias still alive today in California's Sierra Nevada. They grew to at least 70 meters tall, their trunks more than 4 meters in diameter at the base. A lake stretched along the eastern margin of the grove. Insects, fish, mammals, and birds populated the forest floor and the lake's edge. Then, the Gouffé volcanic center, 20 kilometers to the southwest, erupted. A lahar, a volcanic mudflow, swept down through the forest and into the lake. Within minutes, the redwood trunks were buried up to 4 meters deep in volcanic ash and clay. The lakebed itself was sealed under a layer of welded tuff. The Florissant Fossil Beds National Monument was formally designated in 1969. The deposit dates to the late Eocene Priabonian stage, approximately 34.07 million years old, dated by Brad Singer and colleagues in 2002 using argon-argon geochronology on Sanidine. The largest preserved petrified stump at Florissant is the Big Stump, measuring more than 4 meters in diameter. The lake sediments that formed after the lahar preserve more than 1,700 species of fossil insects, plants, fish, mammals, and birds in laminated shales. The Pieridae butterfly Prodrius persephone, described by Samuel Scudder in 1889, is preserved with patterned wings and antennae visible at the millimeter scale. Estella Leopold and Harry McGinnity's 1955 monograph on the Florissant flora documented more than 130 plant species including roses, walnuts, palms, and ginkgos. The Florissant butterfly fossils include the genus Lithodrius and Vanessa amarinda, representing some of the oldest unequivocal Lepidopteran specimens worldwide. Cellular detail is preserved in the petrified redwood wood, including trachite pits, ray cells, and seasonal growth rings, allowing dendrochronology on trees that died 34 million years ago. A 2008 study by Herbert Meyer reconstructed the regional paleoclimate as warm-temperate, with mean annual temperature around 13 degrees Celsius and rainfall about 700 millimeters per year. The fossil deposits indicate that small terrestrial mammals fleeing the lahar were trapped and killed within minutes by the suffocating ash flow. Peter Robinson's 1991 minority view argued that some Florissant insect species had been oversplit, with the actual diversity perhaps half the cataloged total. The famous Big Stump once had circular saw cuts from late 19th century attempts to remove it for display at the 1893 Columbian Exposition. The saw blades broke, and the stump remained where it stood when the lahar swept through. Number 9. The Baltic Amber Forest, also known as the Blue Earth. Of all the death traps in this list, the Baltic Amber Forest is the longest running. The kill mechanism is a single chemical compound. Conifer resin, the resin slides down the bark of an Eocene tree, catches a spider mid-stride, an ant mid-flight, a feather drifting on a breeze. The resin hardens. Over 40 million years, it loses water and polymerizes. It accumulates in the soil. Pleistocene glaciers transport it. It washes up on Baltic beaches today, still carrying the spider, the ant, and the feather, sealed inside. The Baltic Amber deposits are concentrated in the Sambian Peninsula of present-day Kaliningrad Oblast, Russia, hosted in glauconite-rich blue-green sediment locales called the Blue Earth. The amber dates to the Lutetian to Priabonian Eocene, approximately 44 to 35 million years old, based on associated foraminifera and dinoflagellate biostratigraphy. The deposit is the world's largest amber resource, with an estimated 90% of all amber jewelry historically sourced from this single Eocene forest. The most likely resin-producing parent was the extinct conifer Pinus succinifera, although Franz Buyak in 2010 argued that multiple conifer genera, including Cedrus, Pseudolarix, and Picea, contributed to resin production. Kurt Beck's 1986 Minority View proposed Pseudolarix wari as the dominant source, partially supported by recent NMR spectroscopy of amber chemistry. The amber preserves more than 3,500 described species of arthropods, plus feathers, mammalian hair, frog and lizard remains, plant fragments, and fungi, all in near-perfect three-dimensional preservation. The succinic acid chemical signature distinguishes Baltic amber from younger Dominican or older Burmese ambers, with succinic acid content typically between 3 and 8%. Trapped insects often show frozen panic behaviors, including extended legs, splayed wings, and defensive posture, providing direct behavioral data from the Eocene. George Poinar of Oregon State University has been a leading researcher on Baltic amber inclusion since the 1970s and has described hundreds of new species from the deposit. Amber preservation works because the resin is antibacterial, hardens rapidly trapping any small organism that touches it, and progressively dehydrates the inclusion through osmotic water loss. The inclusions include the oldest known fossils of social bees, ants of the modern genera Camponotus and Formica, and the most ancient pseudoscorpion fossils with intact pincers. The Königsberg amber mine, now Yantarny in Kaliningrad, has been the principal commercial source since at least the 17th century. Amber trade routes from the Baltic to the Mediterranean date to at least 1600 BCE, making Baltic amber the longest continuously exploited fossil resource in human history. The forest collapsed as the climate cooled into the Oligocene, with the youngest material around 33 million years, coinciding with the Eocene-Oligocene boundary cooling event. Number 10. The Chinle Petrified Forest. The most familiar forest on this list is also the one that turned to stone fastest. The Petrified Forest National Park in northeastern Arizona was protected as a national monument in 1906 and became a national park in 1962. The fossil-bearing strata belong to the Chinle Formation, dated to the late Triassic Norian to Radian stages, approximately 225 to 207 million years old. The Chinle Formation was deposited across a vast floodplain on the western coast of equatorial Pangaea, traversed by braided rivers carrying volcanic ash and silica-rich water. The petrified logs are dominantly the giant conifer Araucaria oxylon arizonicum, which grew to at least 60 meters tall and 1 to 2 meters in diameter. Recent revisions by Sidney Ash and Rodney Savage have split Araucarioxylon arizonicum into several taxa, including Agathoxylon and the related Pilosilvaxylon, recognizing that the historic species was likely a chimera of multiple distinct conifers. The vertebrate fauna patrolled the river channels and floodplain ponds where Chinle vertebrates went to drink. The long-snouted Phytosaur Smailoscus reached lengths of 7 meters. The armored Etosaur Desmetasuchus, the early dinosaur Colophysis, and the giant temnospondyle amphibian Coscanonodon all left bone in single-death assemblages. Volcanic ash eruptions from the western Cordilleran Arc, just offshore in the Triassic, periodically buried the floodplain in meters of silica-saturated sediment that subsequently petrified the buried logs. Petrification proceeded by replacement of cellulose and lignin by silica polymorphs including quartz, chalcedony, and opal, with the original cellular architecture preserved at the micrometer scale. The brilliant colors of Chinle petrified wood come from trace elements. Iron oxides produce red and yellow, manganese oxides produce purple and black. Chromium can produce green. The Crystal Forest and Jasper Forest sections within the park preserve logs with internal cavities lined with amethyst, citrine, and clear quartz. Crystals. the result of late-stage groundwater silica precipitation. Sidney Ash's career-long research on Chinle paleobotany established the framework for the dominant plant communities. The flora was wiped out at the end of the Triassic by the Central Atlantic Magmatic Province volcanic episode, which triggered global warming and the end-Triassic mass extinction at 201 million years ago. Bill Parker's 2007 Minority View argued for splitting the Chinle Formation into separate land vertebrate biozones that correlate poorly with the dominant Norian-Rachel framework. A 2018 study by Riley Brown and colleagues at the University of New Mexico used high-resolution X-ray tomography to image internal anatomy in petrified wood without sectioning the specimens. The park receives over 800,000 visitors per year and remains one of the most accessible major fossil forests on Earth. Across all 10 of these forests, one principle holds. What kills an animal in a forest is almost never the trees themselves. It is the chemistry of the water and soil they create. Forests modify their substrates through root systems, leaf litter, and chemical exudates, building habitats that double as kill zones for unsuspecting fauna. Stratified lakes adjacent to forests develop permanent anoxia below the thermocline, suffocating fish and insects within hours. Peat swamps generate acidic, tannin-rich water that kills aerobic decomposers and preserves any organism that falls in. Maar lakes ringed by forest accumulate dissolved carbon dioxide and hydrogen sulfide from underlying volcanic activity, periodically releasing lethal gas clouds. Resinous conifer forests trap arthropods, vertebrates, and pollen in viscous tree exudates that harden into amber. Silica-rich hydrothermal systems within forests literally cook plants and animals alive at cellular resolution. Volcanic mudflows triggered by upslope eruptions sweep through forests, burying entire mammal communities in suffocating ash. Hollow plant stems and decaying logs function as one-way traps from which small vertebrates cannot escape. Forests change atmospheric chemistry on timescales of millions of years. They increase oxygen, draw down carbon dioxide, and trigger ocean anoxia downstream. Continental greening across the Devonian fertilized seawater and produced the algal blooms that drove the late Devonian mass extinction. Local kill mechanisms and global climate effects compound across multiple temporal scales. The chemistry of plant decay, especially the production of organic acids, methane, and hydrogen sulfide, drives much of the preservation we exploit today. Permanent water stratification requires both stable temperature gradients and dissolved organic carbon, both of which thriving forests supply continuously. Anoxic mud is the universal currency of fossil forest preservation, from Madygen to Clarkia to Eckfeld. The killing chemistry is rarely lethal to the trees themselves. Trees thrive on the same nutrient cycles that drown the animals. Many of these forests preserved soft tissue, including melanosomes, gut contents, and reproductive structures, providing an unprecedented biological window into ancient life. The most spectacular Lagerstätten of the Mesozoic and Cenozoic are nearly all forest adjacent. Messel, Geiseltal, Florissant, and the Burmese Amber Forests are all included in that count. The death traps required millions of years of continuous accumulation, demonstrating that ecological inertia plus geochemical stability creates the conditions for exceptional preservation. Modern forest edge lake systems in the Congo, the Amazon, and the Indonesian peat swamps replicate these conditions today, producing potential future fossil deposits that scientists tens of millions of years from now may catalog exactly the way we catalog Geiseltal. The molecular chemistry behind these deaths is worth understanding in detail. Anoxia, the absence of dissolved oxygen, halts the metabolic activity of all aerobic bacteria, fungi, and macroinvertebrates that normally consume corpses. Hydrogen sulfide, produced by sulfate-reducing bacteria in anoxic mud, further inhibits any anaerobic scavengers that attempt to colonize carcasses. Low pH in peat swamps below 4 denatures the proteolytic enzymes of decomposer microbes, freezing soft tissue in place. Tannins and other polyphenolic compounds leaching from leaf litter bind covalently to proteins, cross-linking tissues into recalcitrant complexes that resist enzymatic attack. Silica saturation in hydrothermal water replaces cellulose and lignin molecule by molecule, preserving cellular architecture at micrometer resolution. Calcium carbonate concretions form around buried carcasses, sealing them against later groundwater oxidation. Pyrite mineralization, fool's gold, replaces soft tissues in marine and freshwater anoxic muds, preserving feathers, leaves, and arthropod cuticle. Resin chemistry combines deuterpenoid and triterpenoid molecules that are simultaneously antibacterial, water repellent, and capable of polymerizing over millennia. Succinic acid in Baltic amber acts as a long-term preservative, dehydrating inclusions through osmotic water extraction. Then, lignin-rich plant tissues resist decay better than cellulose-rich tissues, biasing forest fossil assemblages toward woody material. Volcanic ash chemistry, particularly aluminium and silica content, accelerates fossilization by promoting rapid mineral precipitation around buried bodies. Thermal stratification of lakes requires summer temperature gradients of at least 4 degrees Celsius per 10 meters of depth. The chemocline, the chemical boundary between oxic and anoxic water, is typically razor-sharp and lethal to any organism that crosses it without adaptation. Decay produces methane and carbon dioxide, increasing buoyancy of corpses and rotating them dorsal side down before sinking. Bacterial mats coat carcasses on the lake floor, locking in soft tissue outlines that subsequent mineralization can preserve. Many fossil forest preservation chemistries depend on rapid burial within days to weeks before scavengers or currents disrupt the assemblage. Different chemistries preserve different tissue types. Silica preserves cell walls. Amber preserves chitin. Anoxic mud preserves chlorophyll. Ash preserves dermal structure. The molecular signature of each deposit allows paleontologists to retrodict the original killing chemistry from the preserved tissue type. Some forests, like the Geiseltal lignite, operated multiple preservation chemistries simultaneously, producing the diversity of preservation styles we see today. The chemistry that kills is the chemistry that preserves, and the better the killer, the more complete the museum that survives it. The relationship between forests and mass extinctions deserves its own chapter. The Gilboa Forest's deep roots drove carbon dioxide drawdown of roughly 1,700 parts per million across the middle and late Devonian, triggering global cooling and ocean anoxia. Algal blooms fed by forest nutrient runoff produced widespread bottom water anoxia that wiped out 75% of marine species in the Kellwasser-Hungenberg events. The Carboniferous Joggins Forest contributed to atmospheric oxygen peaks of 31%, enabling arthropod gigantism, but also setting up the wildfire-driven Carboniferous rainforest collapse. Polyploid lycopsid trees were unable to break down dead wood efficiently, producing the coal deposits that fueled the Industrial Revolution. The end-Triassic Central Atlantic Magmatic Province eruptions, which terminated the Chinle Petrified Forest biotic world, caused a marine extinction comparable to the end Permian in some clades. The Eocene Geiseltal, Eckfeld, Enspel, and Baltic forests recorded the gradual cooling that culminated in the Eocene-Oligocene boundary extinction. The Florissant-Lahar event was local, but is one of the most precisely dated Eocene-Oligocene boundary markers globally. Forest death traps preserve the only continuous record of terrestrial vertebrate response to mass extinction events. Carbon isotope excursions recorded in fossil leaves track. Atmospheric carbon dioxide changes across extinction boundaries. Plant pollen sequences from forest lakebeds document continental vegetation collapse during extinction events at 100-year resolution. The dominance of opportunistic ferns immediately after extinction boundaries, the so-called fern spike, was first recognized in Cretaceous-Paleogene boundary leaves. Insect bite mark studies on fossil leaves track herbivore community collapse across extinction events. The Permo-Triassic mass extinction is recorded in fossil forests by the abrupt disappearance of glossopterid trees and the rise of Lystrosaurus-associated floras. Fossil forest dendrochronology allows reconstruction of growth rings and climatic variation across multi-million-year intervals. The end Cretaceous extinction's terrestrial signature is preserved in the Hell, Creek, and Lance Formation forests of North America. Plant extinction patterns lag behind animal patterns by tens of thousands of years, providing recovery time data for ecosystem dynamics. Carbon and nitrogen isotope ratios in fossil leaves indicate trophic restructuring of forest food webs across extinction events. Modern parallels include Amazon and Borneo deforestation, which release stored carbon dioxide at rates approaching some past extinction events. The fossil forest record demonstrates that vegetation-driven climate change can both cause and resolve mass extinctions. The most lethal landscapes in deep time are often the most ordinary-looking forests of their day, going about the business of growing roots and dropping leaves. What we still do not know about these forests is, in some ways, more interesting than what we do know. The exact parent tree of Baltic Amber remains contested. Multiple conifer genera may have contributed to the resin pool that produced the deposit. The taxonomic identity of Longisquama's dorsal appendages, true protofeathers or convergent scale structures, remains unresolved despite decades of debate. The timing of insect flight origins, complicated by the Rhynie reinterpretation, is currently a moving target. The depth and stratigraphic extent of the Joggins Formation's deeper fossil horizons remain unexplored due to tidal access constraints. The number of separate volcanic events that buried the Florissant forest is disputed. The full chemistry of Eocene Maar lake gas releases is poorly constrained. Modern Lake Nyos provides only a rough analog. The relationship between forest expansion and ocean air anoxia in the Devonian remains quantitatively poorly modeled. The role of cryptogamic crusts in early non-vascular plants in pre-forest weathering is only beginning to be quantified. The complete inventory of Madygen vertebrates is incomplete due to the Soviet-era political restrictions on the deposit's exposure during the Cold War. The molecular preservation pathway producing intact chlorophyll at Clarkia is chemically unusual and still under investigation. The number of plant species hiding in unexamined Baltic amber inclusions is potentially in the thousands. The timing and mechanism of the Carboniferous rainforest collapse continues to be debated by specialists. The end-Triassic extinction's effect on dinosaur diversity remains poorly constrained. The full extent of the Chinle Formation stratigraphic correlation across the western United States is still being refined. DNA preservation in deep-time fossil leaves remains a contested possibility despite multiple high-profile retractions in the years since Golenberg's original Clarkia paper. The full taphonomy of soft tissue preservation in peat swamps requires controlled modern experiments still to be designed. The relationship between fungal mycorrhizae at Rhynie and the colonization of land by plants needs further phylogenetic work. The metabolism and behavior of Chaerovipteryx, the only known hindlimb glider in vertebrate history, remains speculative despite multiple biomechanical reconstructions. The age of the oldest land plants, currently anchored to Rhynie at 411 million years, may be pushed back as new Cambrian and Ordovician finds emerge from molecular phylogenetics. The future of these sites under climate change, flooded mine pits, eroding cliffs, melting permafrost, is genuinely uncertain, and some of them may be lost to research within a generation. Forests have been killing animals for at least 385 million years. The kill rate has only increased as plant chemistry has evolved. The same chemistry that creates lethal water also produces the preservation conditions that give us our most complete window on the past. Every great fossil logger stadia of the Mesozoic and Cenozoic is forest adjacent. Without these death traps, we would have almost no record of soft tissue evolution, behavior, or community ecology in deep time. The Devonian rise of forests fundamentally restructured Earth's atmosphere, oceans, and climate within 20 million years. The Carboniferous coal swamps drew down enough atmospheric carbon to lock in the Permian glaciation. Eocene amber forests captured the diversity of insects at a resolution unmatched by any other fossil deposit on the planet. Modern peat bogs and stratified lakes continue these processes today. Future paleontologists will catalog the Borneo peatlands, the Indonesian mangrove swamps, the Congolese cuvette swamps with the same care that current workers devote to Geiseltal and Eckfeld. The chemistry of forest soils continues to drive nutrient cycling globally. The preservation pathways unique to each fossil forest demand specialized taphonomic understanding to interpret correctly. Our knowledge of ancient communities is heavily biased toward the species that fell into these traps. The forest's ability to kill on local, regional, and global scales remains one of paleontology's most underappreciated dynamics. Modern ecologists are increasingly turning to deep-time fossil forests for analogs for current biodiversity collapses. The deep history of forest killing connects directly to current concerns about Amazon deforestation, peat bog drainage, and continental greening. The Devonian late Kellwasser event's connection to forest expansion has been called the first vegetation-driven mass extinction. The Geiseltal frog orgy layer reminds us that even the most mundane biological behaviors can be preserved across 45 million years if the chemistry of the trap is right. The Rhynie Chert demonstrates that an entire ecosystem can be preserved in cellular detail when conditions align for a few hours. The Baltic Amber Forest reminds us that a single chemical compound, succinic acid, can outlast continents. The Chinle Petrified Forest demonstrates that volcanic ash and silica-rich water can turn an entire river system into stone. These forests are not just biological wonders. They are geological agents, climatic forces, and chemical preservatives whose full influence on Earth's biosphere we are still beginning to understand. The next time you walk into a forest, remember what every animal that walked into these 10 did not learn until it was already too late. The forest is not a backdrop. The forest is the predator, and it has been hunting for nearly 400 million years.